Good afternoon, everyone. Thank you very much for taking the time to attend today. Today, I would like to talk about a new approach to anti-cancer drug discovery using alpacas and AI technology. Let me start by emphasizing that we are not a company that simply produces VHH antibodies. Our mission is to discover novel anti-cancer drugs. As you may know, one of the biggest challenges in cancer treatment is that the target cancer-specific antigens are extremely limited. Currently, antibodies targeting HER2, PD-L1, and PD-1 are widely used in the market. However, as you can see in this diagram, the number of available targets is very limited. At Cognano, we have assembled a unique team of researchers specializing in membrane protein targets and IT engineers. We have focused on alpacas as a source of antibodies, particularly VHH antibodies. Let me emphasize once again, we are not here to talk about producing VHH antibodies. Instead, we will discuss how we identify novel antigens using a unique AI-based approach. As you may know, VHH antibodies are significantly smaller in molecular weight compared to mouse or human antibodies. This gives them the ability to penetrate smaller spaces, which is a useful characteristic. However, we are more interested in another feature of VHH antibodies. Their information can be easily digitized. Here is an example of antibody information. Each antibody's data can be captured as amino acid sequences in digital form, enabling us to obtain vast amounts of data. Using this digital data, we do not need to rely on traditional biological methods to test each antibody. Instead, we employ AI-driven digital analysis, which allows us to efficiently analyze and identify novel antigen molecules. Using this digital processing method, we successfully identified and validated antibodies targeting rare cancers in as little as 6 months. One of our most notable achievements is discovering an antibody targeting triple-negative breast cancer, TNBC. The target of this antibody shows minimal expression in normal tissues, but is highly expressed in TNBC cells. After identifying this antibody, we used reverse engineering techniques to investigate the unknown antigen it binds to. Specifically, we combined immunoprecipitation and mass spectrometry to identify the target molecule. The result? The target is a complex of protein X and protein Y located on the cell surface. Further investigation confirmed that this complex is exclusively expressed on TNBC cells. Using this approach, we are continuously identifying unknown antigens for various rare cancers. Let me reiterate, we are not simply a company that produces VHH antibodies. We are a company dedicated to rapidly identifying novel cancer-specific antigens. Additionally, by leveraging our antigen information, we can develop cost-effective, low-side-effect antibody drugs. This not only offers significant potential for patients, but also increases the probability of business success. If you are interested in our technology, we encourage you to reach out to us for further discussions. Thank you very much for your attention.